Women are from Venus and men are from Mars. That's a well known saying that means sometimes we think and behave in very different ways. But how different are we really, and what would the source of those differences be? Are our brains physically structured differently? How much is genetic? How much is developmental? And how much is cultural? While there is a vast amount of overlap between men and women in terms of anatomy and physiology, understanding the differences can help us make better medicines, improve safety, and design more productive systems. I'm Professor Sunny, and thanks for joining me as I start asking questions. Each society uses various stereotypes to define the typical man and the typical woman. Some of these traits may be anatomical, while others may be behavioral. But even so, each trait can be influenced by biology, culture, and our environment. For instance, while women are traditionally more associated with mammary glands, that may not always be strictly true. The presence of fat tissue in the body increases estrogen levels regardless of a person's sex, and estrogen activates the growth and function of mammary glands, even in men. This means that the mammary gland is increasingly prevalent in boys and men in areas with higher rates of obesity. In fact, the prevalence of the mammary gland in men and boys has increased many times over in recent years. Similarly, breast cancer rates are steadily rising among males. 40% of males can expect to have functioning breast tissue at the end of their life, while two-thirds of men will likely have large deposits of breast fat. Throughout development, males exhibit periods of breast tissue enlargement, including 70% of male babies at birth and 50% of boys at around mid-puberty. At the same time, mammary tissue in women decreases with age, especially upon entering perimenopause. So, which sex is actually more associated with this seemingly simple anatomical trait? The answer depends on a lot of factors and may surprise you. Also, there are cultural influences that can shape our developing biology from a young age. Do parents play rough with their male children more often than with their female children? And does that difference in physical play lead to greater bone and muscle growth? To answer these types of questions, scientists want to remove the influence of culture as much as possible. One way to do that is to look for differences in sexes at as young an age as possible. A recent research study did this by taking scans of the brains of newborns within 28 days of birth. In fact, this is one of the largest studies of its kind because it incorporated 514 newborns. So let's take a look at the data and, more importantly, how the data were collected. This is great that the scientists presented each individual as a visible point on the graph, which is not always done. Because they did this, we can ask important questions about the data, which might not otherwise be available. For instance, we can see that there may be significant outliers within the data, such as some exceptionally large male newborns or some exceedingly small female newborns. Is it possible that these outliers are shifting the entire average? Is it valid to keep or remove these outliers from the dataset? Did these outliers have rare genetic differences that make them irrelevant to the study question? If we had the original data, we could choose a different analysis by asking, did the outliers drive the effect that was seen? This raises one point for me, and that's the issue of data availability in science. Were these data available as a database or spreadsheet with the article, I could do my own analysis and choose my own criteria. And actually, for this research, the data availability is relatively generous. However, it is restricted to individuals that are listed as a primary investigator at a research institution recognized by the National Institutes of Health. So I can't provide you with my own analysis. But the authors did cite the original methods used to capture the data, so we can at least read about how the data were acquired. The neonatal database comes from the larger effort called the Developing Human Connectome Project. This data release and the methods behind it were published in 2022. For projects like this, the researchers focus on collecting well-curated data using stringent methodology. They then publish the detailed methods and release the data for others to analyze. For this project, participants were recruited only from one hospital in London, which is one of the largest and most expensive cities in the developed Western world. So some genetic or cultural factors may not be controlled. Only one imaging center was used, so the standard operating procedures or the habits of that particular center have the potential to introduce skewing to the data. If standard procedures were strictly maintained, any error would be a systematic error that hypothetically affects all participants equally. Because humans are involved, unconscious elements may have caused errors that skew individual results. An example of this might be rushing the scans of participants of a younger age, or being less patient toward the end of the day at a time maybe when parents of older infants might be more likely to bring their children in. If multiple centers were used, at least some of these sources of error would be effectively randomized. A large number of parents of female infants elected to join the study, so there is a self selection bias among parents willing to enter into this study at this specific center. Does this represent a different parenting philosophy for male and female infants? We can't know for certain because cultural influences cannot be effectively randomized when we're only using one location in one city in one country. Also, because you can't ask a newborn to stay still for a 15 to 45-minute MRI scan, these scans were taken while the newborns were asleep, and image correction for any movement was performed later using a computer algorithm to modify the images. To understand the quality of the data, we have to understand these image correction algorithms. And like AI, these algorithms make assumptions about how the image should look and how to transform it to get it there. Those methods are presented in a third paper. During an MRI, strong magnets align the nuclei of atoms, such as hydrogen atoms, in water molecules located throughout the body. This adds energy to the atoms, and when the magnetic field is switched off, the atoms relax back to their original orientation, giving off energy in the process. This energy is released in the form of radio frequencies, which are detected by a receiver. The RF waves are then processed to identify the location of the RF source, which are the atomic nuclei. Rapidly moving the magnet and switching the magnetic field on and off, called resonance and relaxation, allow for building a voxel-based map of RF sources. And the more resonance and relaxation cycles we can run, the better that voxel discrimination becomes. This process typically results in 2D slices due to the circular spinning of the detector and due to the two-dimensional Fourier transform computation to distinguish the signals. A lot of filters and computer-based adjustments are made to the images, including methods intended to correct for motion of the participant in the MRI machine. The image rotation algorithm infers the location of the brain inside the machine by looking for changes in signal sensitivity, kind of like checking what portion of the brain is now closer to the detector. This assumes that the brain is rigid and isn't moving inside the skull. Also, this rotation correction algorithm was validated against images that were artificially rotated around a central axis in the middle of the image, basically using Photoshop to spin the image, not by comparing against ideal scans without motion. This means some amount of error will be introduced into the images, but how much? While these motion correction quality improvements are measured using two seemingly objective metrics, which are called image compressibility and gradient entropy, which basically means sharpness, these two metrics are only valid because they've been correlated to observers' impressions of the quality of the resulting image. None of these metrics measure actual accuracy of the newly created image compared to real ideal scans. So, although image compressibility and gradient entropy are number-based results, I would consider them semi-quantitative data because they rely on a correlation to human judgment, and they don't measure how close the images are to the actual brain being scanned. Ultimately, the results indicate that approximately 70 to 90% of images show improvement in image compressibility and gradient entropy using this method. However, that means that 10 to 30% didn't improve. These images were actually worse, according to the gradient entropy and image compressibility metrics. So, the algorithm to improve images actually made about a fourth of them worse, and that's only measuring things aesthetically by how they look, not based on improving the accuracy to the brain anatomy. Now that we know more about how the MRIs were taken, and now that we know the image correction methods have a 10 to 30% error on their own, let's go back to the original article to look at it more carefully. First, we see that the areas of the brain were identified and labeled by an automated algorithm. The value of this method is that the algorithm presumably treats the same image the same way each time. It's a systematic process that should lead to the same results each time, but it can also introduce errors, such as if a motion correction algorithm adds sharpness where it shouldn't be. Next, we want to know how big of a deal the differences actually are, which is called the effect size. This paper uses something called partial eta squared to do that, and it uses standard values to determine when an effect size is considered important or not. In this study, the smallest effect size that is considered important is a value of 0.01. Here we see in shades of red the regions of the brain that were identified as being larger in females, and in blue we see the region identified as being larger in males. The shades of color used are very dark, but if we look at the partial eta squared values, eta is the Greek letter which is shaped a little bit like a lowercase n, then we see the maximum effect size was only a value of 0.02, which is among the smallest of small effect sizes, considered by scientists to be valid. If we look at the actual numbers, then we see that the effect sizes are mostly 0.01, which barely qualifies as being large enough to be a real effect. Now, let's go back and look at the individual data points that are ghosted into the original plots from the paper. None of the partial eta squared values are above 0.01 when looking at larger scale regions of the brain. And that's not even considering that the image processing steps may have made about 10 to 30% of the images worse than they originally were. I very much wonder what the results would look like if we asked ourselves, is it valid to include these outliers in the data analysis? What do you think? Do you think it's reasonable to ask these questions? Even if there are inherent and biologically determined differences in brain region size, the scientific evidence has for a long time pointed out that brain size is not a determinant of intelligence or intellectual performance. We intuitively know this because otherwise all of the football and basketball players would probably be getting PhDs in their spare time. Typically, what we actually find is that shorter distances and more density in the brain are associated with intelligence, so smaller and more tightly packed brain regions. In general, though, I would say that the results of this complex research are presented in a fantastically clear way. There do remain sources of error which may never be able to be accurately accounted for. Even so, all of science is incremental, even the efforts of people like Louis Pasteur and Richard Feynman. And this research helps, not by reaching a definitive conclusion, but by pushing others to account for even more variables. Thanks for watching. Are you a researcher that wants to make your study bulletproof? Do you want somebody to critically evaluate your scientific writing or to develop your latest grant proposal? Maybe you're a journalist and you want to do more than create a click-worthy headline. Or maybe you're a business leader that wants to find out if you can make your processes more efficient. If so, let me know. I have nearly 2 decades of experience across program management, process development, market research, scientific writing, and consulting. And I enjoy applying scientific and diagnostic thinking to hard problems. And if you just enjoy hearing how science works, then please like and subscribe.